Hi, you guys. Thank you for joining today for this month's expert series talks. Today, we are celebrating endangered species. So the Endangered Species Day was actually last week on May 17th. So we wanted to, bring awareness of endangered species. And so today we have the pleasure of having Amanda Martinez, who actually worked with one of our endangered species called the California least terns. So I'm super excited that she's going to be able to share some of her research that she's done with them. So Amanda received her BS in Environmental Science and Policy from Cal State Long Beach. As an undergrad, she began as a volunteer as a student researcher in the Wetland Ecology Lab. After receiving her BS, her love for wetlands restoration and wildlife led her to continue her journey in the Wetland Ecology Lab, where she received her master's in biology in 2022. Fascinated by birds and the use of umbrella species, she decided to study prey availability of the endangered California least tern. She has worked in the environmental consulting world with Daring Engineering, LLC, and Endemic Environmental Service. She currently works at ICF International as a biologist, where she does restoration monitoring, nesting bird surveys, bat surveys, and construction monitoring. So with that, I will I'll let Amanda take over.
Hi, I'm Amanda, as you all know. And this talk is going to be based off of my master's thesis, just kind of like more truncated version. So for my thesis, I studied prey availability at a variety of California least tern colonies in the Wetland Ecology Lab, like Anita mentioned. So first, a little bit off, what is the California least tern? So it's the smallest of the terns, and for comparison, here is a bird that everyone is usually familiar with, the rock pigeon, and it's about 10 centimeters smaller than, the least tern is 10 centimeters smaller than the pigeon. And so the California least tern is a state and federally endangered species, which means that there is mandated monitoring of the nest site. So here's an example of a one-day-old chick and a chick hatching. Where is this? Here they are. As you can see from the picture, their nest camouflage well with the sand, which means it helps prevent predation, but it has also made them vulnerable to beachgoers, which could contribute to their low population numbers and endangered status. They breed in colonies from May to August each year along the California coast, all the way down into Mexico, usually using areas and sandy beaches adjacent to wetland and riparian habitats. A little bit more about their natural history is that they begin breeding about 2 to 3 years of age. Males will perform a courtship display, which ends in the pair copulating, which means they're a bonded pair, so that pair will stay together for the entire season. Once the pair is bonded, they commence making their nest, or what we call scrape, since it's just digging a little bit of sand, making an indentation. They'll make a couple scrapes, and then the female will lay her eggs in the best one. So they typically have a clutch, or just meaning the number of eggs in the nest, of 2. Here is an example of what a scrape looks like. From 0 to 21 days, both the male and female will incubate the eggs. This is from being laid. After 22 after the egg is laid, chicks hatch. This is an example of a nest that's very well decorated, makes it easier for us to spot. Very pretty. They like select little shells on the beach and add them to their nest. And then At 24 days after hatching, the chick will leave the scrape. After being laid, the chick will leave the scrape. So they're only like 1 or 2 days old at this point. And at 41 days after being laid, fledging begins, meaning the chicks begin to fly. Like I mentioned, the breeding season is from May to August. And for purposes of my study, I divided it into 3 stages. The egg stage, when adults are looking for a mate and eggs are laid. The chick stage, starting when the first chick hatches. And the fledgling stage, starting when the first chick flies and becomes a fledgling. This is an example of what a colony looks like. It's this fenced-in area here. This is Huntington State Beach. This is the Santa Ana River. Talbot Marsh located behind it. And then on this side would be the Pacific Ocean. The overall goal of my study was to understand factors that affect the California least tern and their nesting success, with the ultimate goal to increase population numbers within existing colonies. So I broke it down to potential solutions. So looking at, there's more chicks, more fledglings, we'll have more California least terns. So looking at different parts of their life, we look at, well, we could decrease predation, which is very difficult to do. We already do as much as we can in that aspect, or we can increase habitat quality. That being more plant cover within the colonies or the areas that they're foraging. And then looking at those foraging areas, do they have physical access to food? Are the birds foraging in those habitats? Is there enough food, high enough abundance? And what is the quality of the food? Is it big enough? And then another aspect being the calories of each prey species. For my study, I focused on, and does the timing match? Is there high enough abundance and are they the right size? For my study, I focused on these 4 pink squares right here. And what I mean by the timing matches, here I have a quick graph that I made up, kind of showing the match-mismatch hypothesis, looking at, it's the same idea as the green up theory, which you all may be aware of. Whereas when they're arriving, are the abundance of fish and the sizes increasing with time throughout the season, throughout the stages? Are they going to get what they need? And to kind of like put everything together onto one slide in terms of what I'm looking at what fish are available, what they're choosing to bring back to the colony, and then what they're eating via guano. So the 3 colonies that I studied are these blue circles here. This is the Seal Beach colony, First Basin in Anaheim, and then the Huntington Beach one that I showed you a picture of before. First Basin is a freshwater drainage basin off of the Santa Ana River. The colony is this blue circle, and then these red lines are just to arbitrarily divide it for sampling purposes. And the orange dots are where I was looking at for foraging locations. And that'll be the same for all of the maps. Here at the Huntington Beach colony, like I mentioned before, the river, the marsh, and the Pacific Ocean. And then at the Seal Beach colony, which is a sand-dredged island right here in the middle of the marsh, it has access to the natural marsh, which would be all of this green area. these excavated mitigation ponds, which I refer to as the altered marsh, and the Pacific Ocean. So first I hypothesized that for foraging, they will forage unequally among available habitats. Due to the proximity to the colony, available fish, presence of predators, or humans, which presence of predators and humans was not measured, just looking at proximity and the fish available. So for this study took place from 2018 to 2020, every other week during the breeding season. At each of the available foraging habitats for 20 minutes, I marked down how many foraging flights and foraging dives I observed using binoculars and a spotting scope. At Burris Basin, we found that this closest area here in zone 2 had the most amount of activity, so they seem to prefer the closest spot. Since this is a contiguous drainage basin, it's not surprising that it's going to be the same throughout, so might as well go to the closest place to you. At Huntington Beach, we found that they forage more in the ocean versus the river or Talbot Marsh. And at Seal Beach, they forage more in the altered marsh than the natural marsh or the Pacific Ocean, which may have been just because the altered marsh doesn't, the water level doesn't go up and down as much. So it was always pretty much the same in terms of what they were going to find. And it was fairly close to the colony in relation to the ocean. So overall, it just mattered how close the habitat was to the colony. Nearby open water, close to the colony, and each colony had its own respective location. And so now I'll address the question, whereas if there was enough food, there was a high enough abundance of food. So I hypothesized that in high use foraging habitats, there will be a higher abundance of fish than in habitats with less foraging. And the way I looked at that was via seining at each of those available habitats throughout the breeding season, or cast netting if seining wasn't available just because it didn't work at some places. So we use alternate methods. What we found was that at Burris Basin, it was similar throughout all the zones and all the stages through the breeding season, which is not surprising. You got one pond of water everywhere in the pond is pretty much the same, as long as there's no huge differences in the pond. And the fish we caught were inland silversides, redfin shad, bluegill sunfish, largemouth bass, western mosquito fish, and common carp. At Huntington Beach, I found that the abundance differed by habitats, and the community composition was different in each habitat, which made sense since it had very varying habitats around the colony. The river having more top smell California killifish, rainwater killifish. This is California, this is rainwater, Pacific staghorn sculpin, yellowfin goby, diamond turbot, inland silversides, and oh, I already said that one, yellowfin goby. In the ocean, there was lots of top smelt, halibut, and diamond turbot. And in the estuary, same as the ocean, but adding some killifish and sculpin to the mix. We did find that there were higher abundances in the fledgling stage. And in the river habitat, as depicted by the photograph here. At Seal Beach, fish communities in the altered marsh differed than the natural marsh, so they had different species available. In the altered marsh, we had top smelt, bay pipefish, diamond turbot, different types of gobies, halibut. And then in the altar marsh, not as much variety. Top smelt, inland silverside, to the freshwater, got killifish, diamond turbot, sculpin, and I believe yellowfin goby. Here we also found higher abundances in the fledgling stage and higher in the altered marsh, which may be due to that being an area that isn't really getting affected by the tides as frequently as the natural marsh. And I would like to point out that at Seal Beach, we weren't able to sample the ocean, just it wasn't feasible, so we're missing a little bit of information there. So overall, they preferred nearby open water habitats. Bruce Basin, that was zone 2, Huntington Beach, that was the ocean, and Seal Beach, that was altered marsh. Habitat matters for the types of available fish, showing that in Huntington Beach, the estuary, river, and ocean were all different from each other. At Seal Beach, the altered and natural marsh were different from each other. And at Bruce Basin, everything was similar in terms of what fish species were available and the abundances of those species. So overall, we had high abundance of fish available near each colony in the areas that they use for foraging, so this was a very good sign. So my next part of my objective was I'm going to go to is, was there a high enough abundance of food? I mean, does the size of the fish match? Were the fish long enough, and does the timing match the breeding season? Sorry, getting a little jumbled. So for my third hypothesis, I hypothesized that larger fish will be more abundant in this later in the season relative to earlier in the season, meaning there was more fish available in the fledgling stage than in the egg stage. At Bruce Basin, we found uniform lengths in most years and methods. Fish were in similar length throughout the basin, being around 40 millimeters. At Huntington Beach, we found that fish were similar by stage and habitat. Fish lengths were similar by stage and habitat, but some variability by habitat, with the estuary having longer fish than in the river. Those fish were all around 45 on average. At Seal Beach, there were longer fish available in the fledgling stage than in the egg stage. We didn't really find differences in abundances, but there were longer fish available for them to eat at that time, which confirms a study done in Mexico showing that in early July, they preferred fish around 26 to 50 millimeters. So overall, fish lengths did not change as much as expected during the breeding season. That may be due to needing to increasing sampling prior to arrival and post-migration to really get the full picture of fish growth. Longer fish are available during the fledgling stage, just showing that the fish are growing, which is good. That's what they want to have. They want to have higher caloric content prior to migration. And we did find a difference in length by species, as you can see on this graph. It's not really important what species is, but species is on the x-axis and then average standard length on the y-axis. And what this tells us is that the average lengths of the fish available are being driven by specific species. So if they're choosing a specific length of fish, they're typically choosing specific species of fish. And so we want to make sure that those species of fish are available to them. Next, I'm going to focus on if fish consumed were long enough and if that timing matches. So for my fourth hypothesis, I hypothesized that fish found in guano will be larger towards the end of the breeding season than earlier in the breeding season, and that drop fish will increase in length as the season continues, meaning we will hopefully see the fish growth even in the fish that they're dropping. To answer this question, I do some traditional diet metrics, collecting guano from the colonies, looking at it under a scope, identifying the scaled and otoliths to the lowest taxonomic level, and also measuring those otoliths, as an example here, so I can get an estimate on the size of the fish consumed. And I also collected drop fish as we found them during monitoring, identified them, and I also documented their standard length. So at Burris Basin, we found that they consume longer fish later in the season in their guano. Fish longer in chick. They consumed longer fish in the chick and fledgling stages than in the egg stage, those being around 30 millimeters. For drop fish, overall, the lengths were similar throughout the breeding season. They were 60 millimeters or longer. However, this may be because at this colony, there are a lot of other seabirds nesting on the island, and they were all larger than the least tern and capable of eating bigger fish, and we're kind of messy, dropping fish everywhere. At Huntington Beach, we found that in their guano, that they consumed longer fish in the egg stage than in the chick and fledgling stage. This may be due to during this time in the egg stage would be the time where they're trying to the males are enticing the females, and they might want to select the biggest, juiciest fish to present to her, and then consequently they're eating those fish, and so that may skew the data a bit. And then for drop fish at Huntington Beach, they were similar throughout the breeding season. And a good point was that they were longer than the consumed fish, which seemed to be a trend so far. At Seal Beach, guano was longer in the egg and fledgling stages. Than in the chick stage. For the drop fish, we found that it was similar in all stages, all around 60 millimeters. So overall, the California least term consumed longer fish in the egg and fledgling stages, with the average fish consumed being around 10 to 50 millimeters. Drop fish were similar in lengths, typically longer than 60 millimeters, and I would like to point out that birds usually, especially a small bird like this, they can only open their mouth so wide. And once fish tend to get seems to be over that 50 millimeter mark. They seem to be too wide for them to swallow, and that might be why we're finding them on the ground and not smaller fish so much, since if you ever watched a bird colony or an adult seabird try to feed its chick, it definitely keeps trying until it can't at all or will not eat that fish. It will keep trying to give the chick food. And also in the dropfish, same as within the seining data, we found that there was a difference in length by species. So they were selecting specific species and they all tend to be around the same size for each species. So just a quick overview of going back to that integrated trophic support, just so you could see the fish that we found in each of these sections. So this here is for Burris Basin, and from the available fish, all those same fish were brought back to the colony, and of those fish, all of the fish were consumed. That's what the highlighting denotes. Just in this case, it kind of worked out that it was in every stage that we looked at. At Huntington Beach, there's a little more selection from the available fish here on the left-hand side of the screen. There was actually some fish that they did not want to eat. And then from the drop fish, these are the fish that were also in the available fish and in the guano as well. And the last colony, Seal Beach, a little more selective here. So from the available fish, we only found 4 fish, 4 of those species in the dropfish, and 4 of the families in the guano. In conclusion, open water habitat in close proximity to the colony is very important. Habitats that have an abundant slender body, appropriate sized fish seem to be what they prefer to fish in. And multiple metrics are key to catching future changes. Although predation remains the greatest threat, we can't really do much more about that at the moment. The next direction would be to connect this data with the nesting success at each colony. So why should we care? We should care about this project and about this specific endangered species because California least tern lives in coastal areas typically adjacent to wetland habitats. Coastal habitats have been greatly negatively affected by recreation, human development, to the point where we're endangering the animals that need them in order to survive. Since the least tern is an endangered species, it acts as an umbrella species to all its nearby inhabitants and protecting them all as well. Every critter is interconnected through food web interactions, and if one piece were to be taken out, it can cause cascading of negative effects on an already struggling ecosystem. Also, mandated monitoring of their nest sites is government funded, and as taxpayers, we should care about how effective this program is. That's where I come in attempting to improve monitoring strategies by filling in the gaps in knowledge that can aid land managers' decisions on how to help the California least tern population. So I want to thank all the people who helped me with this project with tons of field work and lab work, all the volunteers. There's definitely way more people than I was able to put on this list, as well as all the land managers and funding, of course. Just want to shout out to them. And are there any questions?
Also, that is a very cute picture, by the way. So cute.
It's my favorite one. That's why I put it here.
He's like his little running.
Yeah, Dale Halliday has great bird photos. He has a Flickr account if you're interested.
Oh my God, so cute. Um, so I so great talk, by the way. I did have a question. It's kind of interesting how like different sites their dropped fish when you looked at the guano was very different depending on the stages. Because like I think like what Seal Beach there was more in the eggs and the fledgling the eggs in the fledgling stage.
I
But
believe so,
then
yeah.
like in like one of the other sites, it was like either it was like just a fleshly stage or all of them. So I thought that was kind of interesting. And why would you do have a theory why it changes per site?
I think a little bit it depends on let me go back to those. Might be a little, let me think about it. Oh God, I forgot I have all these animations, so it'll take forever to go back. It's hard to say. Like each site is just totally different. That's why I wanted to compare three different sites, because if you just look at one site, they all have varying habitats and everything's completely different around them. So that's part of the reason I wanted to do this. We're still trying to figure out like why those things were so different. And it also could be just to get the sizes of the fish they ate, you need to find a lot of otoliths, which are kind of luck of the draw. So sometimes it's just we just didn't have a lot of data. And so maybe that result would change if we went through more samples. But yeah, the samples take quite a while to go through. So we went through as many as I went through as many as I could. Um, so that's my biggest guess right now is that there just needs to be more data overall, but just the varying habitats makes a huge difference because they don't prefer the same places no matter where they're at. Like I thought maybe at Seal Beach, they would also prefer the ocean, just like they did at Huntington Beach, but they didn't. And so I was like, okay, so they're just eating different food in general. And then you have to factor in that the otoliths get eroded as they go through the digestive tract, which kind of affects how big we're estimating the fish to be consumed.
Mhm. Okay, thanks.
Calvin
Calvin
raised his
has a
hand.
question.
Hello, Calvin.
Of course I do. Yeah, great talk. So my question relates to it kind of relates to that sort of selectivity in terms of where they're foraging and what they're consuming. Did you happen to look at water clarity? Like, did you measure that in some way, whether it was with Secchi discs or turbidity meters or anything like that, given
Um,
that they're a visual predator?
no, that's a really good point. We didn't look at water clarity or anything. That was something that like we thought about at one point, like maybe putting some loggers out there and getting some abiotic data, but um, as you may know, it's quite difficult to work with a lot of different land managers and get everything done in time so it's uniform. So we didn't end up focusing on that. We're like, we'll just do what we can, like what we're allowed to do. But I think that would be a good point because I think that'd be really interesting just to look at like the first, I don't know how many centimeters it is, but like they don't go very deep. They plunge in and they're just on the surface of the water. So getting that data would be really helpful in comparing it at each of the habitats and seeing if the water is clear in those habitats they prefer. Because I assume the natural marsh, at least for Seal Beach, with the water level constantly changing as the tide goes in and out, it's not as easy to see fish as it would be in the ponds, which is nearby. And so that could be why, like, oh, it's easier to fish here. I'll just fish in the ponds. But yeah, that was a good point. Thank you.
I don't think I have any other questions unless you guys have any. I also did notice that you had a picture of me and Chloe singing. I didn't know that that photo existed.
Oh yeah, that's the go-to one. I like it. I had some with you guys were in the mud and stuff too, which I really wanted to use, but I was like, this is what the typical seine looks like, so I used that one instead. It was that same day, I'm pretty sure. It was just at a different spot.
Oh.
You guys were crawling. It was great.
Oh, I remember that
I,
one.
I like recorded, I made a video.
Yeah, I remember the tide was super low and we had to army crawl across.
Yeah, I was
So why didn't
surprised
you
we
show
even
that
caught
video?
anything. Oh, it's buried in the files.
Somewhere.
I look at it for my own pure enjoyment sometimes.
How many, actually, I just remembered. Okay, how many like odorlists did you collect? Or was it just like kind of like random? It's like whatever you find.
Oh yeah, it was whatever I found. It's pretty random, like so I don't even know. I mean, I probably collected at least like at least over 100 or so. Like that's my just pure guesstimate. Like no, I haven't looked at any of those numbers. But some sites it would be like I had the little pill bottle and otoliths were grouped together by sample and like little pill capsules, and sometimes that bottle would be completely full, and other times it'd have like two, and I was like, oh, so sad. But yeah,
Were you
that's what
able
I
to
mean
like
too. Luck of the draw.
distinguish like the guano from like other bird species? Because like I think like you mentioned like especially like Barris Basin, there was like some other fish, I mean other birds around the area.
Yeah.
So like can you determine who's like, oh, this is a least tern guano compared to like a different species?
So for that, at that colony, I specifically collected like directly around the nest and
Okay.
where I saw them poop. But it also like they're a lot smaller than the other birds, bigger poops. So kind of avoid the bigger poops if it doesn't look like a tern poop. Once you see a lot of poop, you know the difference.
So you're an expert in poop.
Just in the seabird poop. I did observe a mourning dove poop, which was wild. It's a spiral. It's like just like this little brown skinny spiral. We were like, what is that? And we were like, oh, it's just poop.
So that's a skill that you never thought you would have, huh?
I never
It's definitely a
thought
CV
I would have
builder
it.
right there.
I can spot the poops.